We'll get started in just a minute. All right, welcome everyone to our annual research day. My name is Barbara Bendlin, and I'm a professor in the Division of Geriatrics and Gerontology. And I lead the Wisconsin Alzheimer's Disease Research Center research education component, which trains the next generation of dementia scientists. So we're happy that you have all joined us today to learn about the phenomenal science, groundbreaking work that's going on right here in Wisconsin, as well as beyond. This is really an exciting time to be engaged in Alzheimer's disease research at a time when really our decades of research is now impacting people who are affected by Alzheimer's disease and related dementias. So our meeting is going to, just some logistics, our meeting is going to happen in this room as well as outside in the forum. We are going to have lots of opportunity to hear talks here in the forum, but also have time to engage with one another, do networking, and we are also going to have a social at the end of the day. So stay tuned for that. Backed by popular demand, we are also going to have some raffles throughout the day. So you must be present to win. We've got some great prizes. We've got books, we've got squishy brains, we've got mugs. So no t-shirt gun this year, but that will come in a future year. So also keep out your eyes for QR codes. We're going to have lightning presentations today, as well as posters. We invite you to tour the posters, pick your favorite one. We want you to actually register your votes for your favorite poster, as well as your favorite lightning presenter. So keep an eye out for that. We have a number of special guests here today. We're going to hear some really wonderful presentations. We're going to learn about what everybody is doing on campus, have an opportunity to meet new people, and promote collaboration. And it's really wonderful to all be here together. This is really an opportunity to hear what your colleagues are doing and to celebrate all the great science that our trainees are doing as well. If you're joining us via live stream, thank you for joining us virtually. You will be able to use the Q&A function on the live stream. Please just drop any questions that you have into the Q&A, and we will be monitoring that. So thank you if you're joining us virtually. I also want to thank, of course, everybody that made this day possible. First of all, first and foremost, Cheryl Spenceley. Thank you, Cheryl. She's in the back there. And as we know, none of this would be happening without Cheryl. So thank you so much, Cheryl, for all of your tremendous efforts to make today successful. And I also want to thank the Discovery Center for hosting us. Thank you for the IT team in the back. And thank you for all of the volunteers that are helping us today, all of the poster judges. We really appreciate it. All of the faculty who are here to support their trainees, and all of the volunteers who've really been working hard to make this happen. So thanks, everybody. I am now delighted to introduce the Associate Dean for Gerontology and Founding Director of our Alzheimer's Disease Research Center, Dr. Sanjay Asthana, to deliver highlights for year 15 of the Alzheimer's Disease Research Center.
All right. Good morning, everyone. How y'all doing? Great to have you all. Welcome and thank you all for being here. Dr. Bendlin mentioned my role here is to give you a very brief overview of the Wisconsin ADRC. And our sincere hope is that as you move around and talk to people here, you might be able to establish some collaborations with scientists sitting here, because we have audience from all over the campus, and there's so much of exciting work going on here in Alzheimer's that it'll be great if you get a chance to talk to people and perhaps develop some collaborative projects. Okay, so this is just a reminder that how big the problem of Alzheimer's disease is and the devastations that are associated with the disease. So in the United States alone, currently we have about 7 million Americans who have the disease. And by 2050, that number is going to increase to almost 14 million Americans unless we find some effective treatments or preventive strategies for the disease. In Wisconsin today, we have about 110,000 people who have the disease. Just in the next 2 years or so, that number will go up to 130,000 people with the disease. And we all know that this disease just doesn't affect the patient. It affects the families, the care partners, and also the society at large. So it's very, very devastating socioeconomically and also on the family. Next slide, please. So the Wisconsin ADRC was funded by the NIH in 2009. So we are in 15th year of our funding right now. And the good news is we've been told that we'll be renewed for another 5 years by the NIH. And this is competitive funding. We are delighted that that'll happen, and we're expecting the contract any day. Thank you. Thank you. And again, this is the result of collective efforts and some very interesting research that continues to come out of the Wisconsin ADRC. So there are 33 fully funded ADRCs and 4 exploratory centers. So you see the light blue color. These are the 4 centers that were funded by NIH with the hope that they will, with some initial funding, expand into the full-blown Alzheimer's disease. Next slide, please. So this is just a brief summary of just in the past 1 year, or in fact, in the last funding cycle, I should say, we accomplished quite a bit. We completed over 200 research studies. We published over 300 papers. Since inception, we have published over 1,400 papers from our center. And also, we were able to create a team of 33 core scientists. These are scientists of all backgrounds, and they come from all colleges, schools, centers, institutes across the UW campus. The center really is a campus-wide program. And in addition to 33 core scientists, we have 79 affiliated scientists, again, spread all over the campus. And each year, we really believe in training the next generation of scientists. And each year, we train about 100 learners of all backgrounds who have all kinds of research interests. And that training is dominantly done by Dr. Barb, Bendlin, and program. And Cheryl, please join me in applauding the amount of work that goes on in Dr. Bendlin's program to train these trainees. Next slide, please. So one of the distinguishing feature here that the ADRC has, we support a large cohort of very well characterized participants who are at risk for the disease. And currently between RAP, and you all have heard about RAP, the Wisconsin Registry for Alzheimer's Prevention, and the ADRC, we have over 2,300 participants who were recruited when they were in their middle age. And it looked like that risk factors at mid-age may be most important in terms of future development for the disease. So we have a large cohort that has been well characterized. We have extensive prospectively collected data on these participants, and they've been followed almost up to between 10 to 20 years. So we have 20 years of data on many of these participants, and these data are shared locally, nationally, and worldwide. And they have resulted in really cutting-edge findings in many aspects of Alzheimer's disease, but we have some distinct strengths here, and I'll, in the next slide, just identify those strengths for you. And as I mentioned earlier, we also have trained a number of people who have utilized our data. one of the unique strengths here is the community engaged research. What that means is that our investigators staff go all over the state of Wisconsin, and they communicate with the especially underserved, underrepresented group populations, and share with them how important research in Alzheimer's is. And as a result, we have one of the largest cohorts of African Americans and American Indians in the Wisconsin ADRC across the country. And again, this is the result of enormous efforts put in by staff. Next slide, please. And as I mentioned earlier, we do have some distinctive strengths here at the ADRC, which are recognized internationally. So one of them is preclinical biomarkers of Alzheimer's disease. We are among the first programs to focus on this area of research, and Dr. Sterling Johnson and his team, they're world-renowned. Dr. Hendrik Zetterberg, I don't know if he's here. Hendrik? Yep. Dr. Zetterberg is a world-renowned scientist who really initiated the field of blood-based and CSF biomarkers. It is a privilege for us to have him as a faculty here in the ADRC. And if you get a chance, please talk to Dr. Zetterberg. He is an amazing scientist in this field. Also, our next strength is health equity and disparities. As I mentioned, we have a large cohort of African American, American Indian participants, and we have published numerous papers. Dr. Carrie Gleason, she's here. Carrie, are you here? And there she is. Dr. Dorothy Edwards and Amy Kind have really focused on health disparities research, and we better understand how the disease differs in different races and ethnicities, and also social determinants of health. It is very clear that various social determinants of health, poverty, education, employment, your social network, all those kind of things significantly affect the pathobiology of Alzheimer's disease and other disorders. And Dr. Amy Kind is the world leader here, and she's brought attention to how important it is for us to study, in addition to biomarkers, the social determinants of health, which occur over the life course. They just don't happen, for a few years. Really, they start to work from the moment we are born all the way to our later lives. And of course, I mentioned about the community engaged research. Next slide, please. We have one of the largest biomarkers program within the ADRC network, and we have multiple labs and we have multiple platforms and equipment through which we can assay all kinds of AD biomarkers right here locally. And once again, thanks to Dr. Zetterberg, to Dr. Johnson, and Dr. Rachel Wilson, if she might be here. Under their leadership, we can assay all kinds of biomarkers in the CSF and blood right here at the Wisconsin ADRC. And this program is expanding rapidly, and a lot of investments are being made so that we can do actually discovery and innovation and discover new biomarkers, especially in blood. Next slide, please. And this is just in the last year, in fact, year 5 accomplishment. We have now over 1,100 participants who are participating in the ADRC studies, and they provide all kinds of data. And about 80% of them do 2 or 3 or 4 studies, not just one study. And we have a large brain bank. We distribute brain tissue worldwide. Our autopsy rate is about 60% and 60%, and the consent rate is about 63 to 66%. Next slide, please. Clarity grant, you probably all know, Dr. Johnson brought in the largest grant ever awarded to UW-Madison from the NIH. And this grant is focusing on identifying multiple pathologies that exist in people who have a diagnosis of Alzheimer's. So when we do autopsy on those people, it's not just Alzheimer's pathology. We see multiple other co-pathologies that exist, and they contribute to the symptoms and progression of the disease. And Dr. Johnson, in collaboration with almost every ADRC in the country, is trying to identify biomarkers and imaging markers for these coexisting pathologies. Once we do that, targeting those co-pathologies might be an effective strategy to manage dementia, not just due to Alzheimer's, but other diseases. Next slide, please. And this is another study. Dr. Carrie Gleason is leading a nationally renowned study called AA-FAME. Do I have 2 minutes? Have I gone over? Am I good? Okay, we have last 2 slides here. So Dr. Gleason is focusing once again on understanding the biology of the disease in underserved populations, mainly African American and Native American participants. So AFAM is African Americans fighting Alzheimer's in midlife. This has over 600 participants carried data gathered, right? Somewhere between 500 to 600 African Americans. They're being followed prospectively. They are giving us biospecimens, extensive research data, and those data are being published. So these are some of the first data that will show how the disease differs in this population. Next slide, please. And finally, there's another grant given to us by NIH, a large grant, which is investigating the sociobiological basis of Alzheimer's disease and other dementia. I mentioned social determinants of health, and this study is evaluating participants in the Wisconsin longitudinal studies, which are high school graduates of Wisconsin of 1957. They've been followed for 67 years by UW-Madison, funded by NIH since 1999. This study has been funded since 1999. And we at the ADRC came in, got a grant from NIH to establish dementia status in these people. And the renewed grant will help us collect biomarker data on these participants as well. Next slide, please. And they are some of the our new trainees here that we have selected, and some accolades and awards given to a number of our faculty. I won't go into detail, but you'll get a chance to look at all the recognition awards our faculty have received over time. Next slide, please. We provide support to young faculty to do some innovative research they call DPs, or developmental projects, and we give them about $150K over 2 years, and they have conducted some breakthrough research through these funds. Next slide, please. Once again, this is the Rec Scholar, where we support some young investigators and help them attract NIH grants, generate some pilot data. So here are two of the living examples of these successful trainees. Next slide, please. And finally, Dr. Nate Chin. I think many of you have met him. Dr. Nate Chin has created an internationally renowned podcast program called Dementia Matters. And these podcasts have been listened to worldwide. There are over 175 episodes of these podcasts. If you get a chance, listen to some of them. They've been downloaded in almost 130 countries and over 650,000 times. So these podcasts have been heard throughout the nation and worldwide, and they really provide you basic information about aspects of Alzheimer's disease. I think this is my last slide. So I want to thank you all for being here. I might have gone over a few minutes. There's so much to describe and share with you. We hope you enjoy the day. Thank you.
Thank you, Dr. Asthana, for those wonderful updates. I'd now like to invite the Associate Director of the Alzheimer's Disease Research Center and leader of the Wisconsin Registry for Alzheimer's Prevention and PI of the Clarity Grant, et cetera, Dr. Sterling Johnson, to introduce our first featured speaker. Dr. Johnson, thank you.
Good morning, everybody. It's my pleasure to introduce our first keynote speaker today, who's Dr. Kris Saha, who's an associate professor here at the University of Wisconsin. And let me just say a few words about him. Dr. Saha is, as I said, an associate professor here. He's the Retina Research Foundation Catherine and Latimer Murphy Chair, an endowed chair to support his work. He's appointed in the Department of Biomedical Engineering and also the Department of Medical History and Bioethics. Dr. Saha was trained at the University of Berkeley, where he got his PhD. He went on to do postdoctoral training at MIT, at the Whitehead Institute, and also had a joint appointment as a fellow in bioethics. So he's approaching the world of of chemical engineering and genome editing from not just a scientific point of view, but also understands and is thoughtful about the policy and the ethics of doing this kind of groundbreaking research to affect the lives of patients. He's had a number of highlights, a number of awards to highlight, including he's been elected as a fellow of the American Institute for Medical and Biological Engineering. He is the co-chair of the steering committee of the NIH Somatic Cell Genome Editing Consortium, which is a groundbreaking consortium that has ample resources behind it to understand genome editing. He's had a number of career awards, including the Gund Harrison Scholar Award, the NIH Maximizing Investigators Research Award, or MIRA. He's had a NARSAT award from the Brain and Behavior Research Foundation, an NSF Early Faculty Career Award, also one from the Allen Institute, and I can mention one or two others here. Also one from SAGE BioNetworks Young Investigator Award. So you get the idea that he's being recognized for his potential back then when he was a young investigator, but now for the impact he is making. He has over 89 publications. He's funded from NIH, including providing leadership to this consortium, this genome editing consortium that I mentioned, which is really a one-of-a-kind consortium that's focused on finding therapy for diseases through genome editing. And it was a pleasure to talk with him a little last night and get a sense of why he does this. He is doing this work so that he can affect the lives of patients, and that is so that's central to what we all do. And it's so it was just great to connect with him on that level. That's something that we have in common, that we want to affect the lives of our participants. Well, let me stop talking and turn it over to Dr. Saha.
Okay, well, it's wonderful to be here, and thank you, Dr. Bendlin and Dr. Asthana and the organizers for inviting me to share our work here with you today. And I think the focus on early biomarkers is something that's really crucial to the development of some new advanced therapies that I'll be talking about that use CRISPR, because we want to be able to intervene at the right point and safely and with a lot of efficacy. And it's, you know, in a disease like Alzheimer's or dementia, this is very important to get right. And look forward to hearing from many people in the center about all of the great work that is going on here at Wisconsin. This work is really a collaborative story. I think many labs here benefit from the really rich environment. My lab is here upstairs at on the 4th floor, and we've had the fortune of having students from engineering, but also from biology and molecular pathology, neuroscience program. So it's been really a lot of fun to do this work. So what you're seeing here on this video is a recent video of some senescent cells tagged in green that are being recognized and attacked by a genetically engineered T cell. So this T cell has been CRISPR'd, and I'll tell you what that's about, to recognize some surface markers on the senescent cell. So once it finds it, it grows and then starts eliminating the green cells. And I'll tell you how that process works. So just a review, genome editing, the concept is to fix typographical errors in the genome. And so this T base is a pathologic single base mutation in 1 in 3,000 bases in our genome. If you were able to fix it precisely to a C in the right tissue, you would be able to address and treat an inherited retinal disorder that causes defects in central vision called Best disease. What patients see in that disease is shown here, where you have peripheral vision intact, but really your central vision degrades over time. And so we're trying to address this by injecting some genome editors into the back of the eye. These genome editors really work using a multi-component system. What you see here in orange is a short piece of RNA called a guide RNA. It can be easily synthesized, and then an enzyme protein called Cas9. They come together, and sometimes there's an additional nucleic acid in teal that's DNA. So if you're able to deliver the RNA and the enzyme into the nucleus, it will scan the genome in teal. And once about 20 base pair target sequence matches the sequence on the RNA, which is in orange here, it will trigger and give time for this enzyme, Cas9, to cut both strands of the genome. And you'll see that in this cartoon happening here. Once that happens, the cell takes over. So there's DNA repair proteins that recognize broken DNA strands. And if you have a template around, like this blue DNA, it will use that to write in a sequence where you want and to edit precisely a piece of genetic code. So there's been many types of these editors applied really in therapeutic products. So, I mean, it's really exciting to show this slide now, 12 years after I started my lab here, that there's actual products that use CRISPR. The one that was approved in November was called Casgevy, that takes bone marrow, uses CRISPR to modify a portion of the regulation of hemoglobin to turn on fetal hemoglobin in adults to treat sickle cell disease. And so those are cells taken out, edited, and put back into a patient. CAR T-cell therapy is also a form of gene and cell therapy that are essentially combined, where you take out T cells, use genetic engineering to write in a chimeric antigen receptor, one of those receptors that I showed in the red cell at the very beginning video, to recognize a cancer cell and to eliminate that. There's 6 approved products there. At the moment, they don't use CRISPR, but there's many programs underway to use CRISPR there. And then there has been a clinical trial, not an approved product, where they treated a number of patients. One is Michael Kalbrer, who got an injection of a viral vector that encodes CRISPR into the back of the eye. And they did see some reports of efficacy, even though it was a safety trial. And that was just reported a few years ago. One of the major concerns, and I think this is where our mission intersects in part with the ADRC, is that access to these therapies is very limited. The costs of these types of therapies range from $400,000 to $2 million per dose, or per eye in the retinal case. So these are places where we think we can develop new strategies to make these types of products, perhaps at a lower cost of goods standpoint. So the consortium that Dr. Johnson mentioned, that I had the fortune of leading for 3 years, was this consortium funded by the Common Fund at the NIH. It has developed a number of delivery strategies to get those enzymes and RNA into the nucleus. They've also developed a number of different editors that can do not only cutting of DNA, but nicking and base editing of DNA. And then some of these genomic targets are not found in animals, they're only in human cells, making tissue chips, organoids, brain tissues in a small format that are human is also a focus of this consortium. So we've been working with a number of editors. The one that I'll mostly talk about today is Cas9, which is actually some people call the most boring editor out there because it was the first one discovered in 2012. But it's actually still very useful and works really well if you are able to control it well. The lifetime of it within cells pretty precisely. We also use some base editors that are down here, where you can precisely edit the T to a C that I showed at the very beginning. And then prime editors as well have been used where you fuse a repair template to the actual enzyme and guide RNA complex. Those have been used in the brain, and I'll show you one example of that. This is part of a toolbox that's been really rapidly expanding. Earlier this week, there was a story of new types of enzymes that have been developed through AI. And so this is a phylogenetic tree, and Cas9, the one that we use, is from one bacteria, Streptomyces pyogenes, that's here. But you can see in evolution, there's many other natural types of Cas9, and actually what this company Profluent was able to do was to make about 95 more, expand the types of editors to quite significantly to make a lot more diverse types of enzymes. So, I think I'll show you all the progress with what we're doing with the very first one. You can imagine what you'd be able to do with some of these other editors as well. And so the last introductory slide here is to say we are moving these types of projects towards the clinic. The project that's being advanced by this consortium, I guess, to get into patients by 2028 is this one targeting the eye. I won't go into detail of this, but I think a lot of the know-how about trying to do this at the appropriate scale and quality and measure off targets are things that would help the general campus community as well as the field in moving more gene therapies forward. So we're happy to do this here in Madison and share all of our know-how as we do this over the next few years. So the rest of my time, I'm going to focus on two stories that are focused more on Alzheimer's. So the first one is what I just showed you, which is mostly direct injection of a formulation into a tissue. Like the back of the eye, in this case, we're targeting parts of the brain. And then the second story will be the engineering of T cells outside the body. That is a story where we've engineered T cells to target senescent cells by CRISPR. So I think I thought I'd tell you why direct injection of CRISPR into the brain is not a crazy idea. One of this is that there's been various variants that have been identified through really phenomenal clinical work. One of this is the Christchurch mutation about 5 years ago in ApoE3. And you can see that here in the homozygous A switched from a C. And if you're able to make this type of switch is thought to be potentially protective against Alzheimer's disease. And so there was a team that developed genome editors that would target this specific C base and make it into an A using prime editing. And they were able to do this at fairly good efficiencies. About 30% of the cells that got the editor made this very precise switch. This is, of course, in mice. And so there's a lot of work thinking about how you would move this into patients. What we've targeted instead was APP. This is work with Subhajit Roy when he was here at Wisconsin. And we looked at the very C-terminal part of APP, which actually gets cleaved by a number of secretases. Base 1 is one of them. You can see it cleaves up here. And there are these sites that are good target sites for Cas9 that have this proto-adjacent motif called PAM, TGG, these types of sequences that make it good for CRISPR editing. And so we designed a strategy to cut off essentially the last portion of APP. And through this work, we've been able to see that when we do that editing, it shifts this balance towards this non-amyloidogenic pathway. And so rather than seeing this type of cleavage, we're able to now make this edited allele insensitive, and the protein product cleave differently. So Subhajit's lab, and namely Brent Alston there, a postdoc who's led this work, has made germline edits in a mouse model that has this human allele, in addition to 3 familial mutations that help drive AD pathology. And they end up getting plaques during the first few months of life. They have neuroinflammation, so they model aspects of AD. And what we ended up doing was to monitor plaques here at after a year or two. And certainly when we knock in that diseased allele and then stain for A beta, you can see these little spots here that Subhajit tells me are plaques. And in the one that we've edited, we see much less. And so that's what the del5 and the insertion T are. Those are CRISPR edits exactly at the C-terminus of the APP gene, this disease gene, that end up reducing very dramatically plaques in this model. We also see a number of functional and behavioral changes. This one is a water maze test, and in this case, this was done in a way that is more relevant to what you would try to do in a patient, which is to inject CRISPR into an adult mouse, well, about 1.5 months old, and then we evaluate about 11 months later. And so what we see is that when we load this editor against APP, which is in red, versus load the editor against a irrelevant gene that's not in the mouse genome as a control, we see much more time investigating these type of novel objects in this memory test. And so we think we also see a lot of reduction in plaques, as I mentioned. And what we're trying to do now is to look at the cells within these brains that have been edited in much more detail. So a postdoc in my lab, Dr. Kirsten Gimsey, has been doing single nuclei transcriptomic analysis of these brains that have been essentially shipped from San Diego to here. And this is a single map of 19,000 cells, and we're able to identify really a nice representation of cell types in the hippocampus. And what we find from this analysis, and what she's found, was that within microglia in particular, the diseased associated microglia, when you do the editing, which is in green here, compared to the diseased one, which is in red, you see a marked reduction in some of these genes that are expressed specifically in those disease-associated microglia. She has another story about oligodendrocytes, and she's also looking at neurons. But we're starting to get a sense of what this type of CRISPR editing could be doing over a month to year timescale when CRISPR has been administered in a living intact tissue. So the second story I want to quickly go through in my remaining time is rather than injecting CRISPR into the brain, we take out T cells and use CRISPR to write in a new sequence. And the sequence we're writing in is this long set of protein domains that have been fused together called a chimeric antigen receptor. And so what you see here in red is one of these cells that have the chimeric antigen receptor and is really sticking on and recognizing the cell surface of a dysfunctional cell. In this case, it's a tumor cell, but there's other dysfunctional cells, really any specific antigen that you can raise this receptor against will confer the immune cell from first recognizing, binding, and then attacking that cell. And so this is standard single chain variable fragments from an antibody. And the intracellular part of this is T cell receptors. This can be done in the brain and within patients. So earlier this year, there was really exciting results from work in Philadelphia and Boston that treated 6 patients that had glioblastoma. You can see the mass here before treatment, and then after treatment, it disappeared. This reduction was pretty marked, and they were able to deliver a high amount of cells intrathecally. So this at least gives some faith in that we can start to explore whether these T cells cannot recognize per se cancer, but perhaps other dysfunctional cells in the brain. And so we can make these cells using CRISPR really efficiently. I won't go into the details, but happy to talk about this in questions. And Lauren and David from my group are presenting posters. What we do is we knock out the T cell receptor. This alpha chain of the T cell receptor can be used to drive the synthetic receptor. And so over here, this is a stretch of DNA that we encode our chimeric antigen receptor. And this can be now done fairly efficiently and very, very quickly. And what we decided to do was to target senescent cells within the brain, but also these cells could be used outside the brain as well. The reason for looking at senescent cells is that there's been some prior studies that suggest that clearing senescent cells can help neurogenesis and some aspects of pathology. You can see the increase of a senescent marker as you add more of this A-beta increasing concentration. And then as you increase in age, you see an increase in senescent cells around some of these problematic areas of the brain. So what we ended up doing was targeting this molecule on the cell surface called a urokinase plasminogen activating receptor, or UPAR for short. And it gets turned on in senescent cells. It also gets cleaved to make a secreted form of UPAR. We don't really do much with the secreted form, but it has been used as an interesting biomarker for all sorts of purposes. So if that's something that we can measure through some of the samples, we'd be very interested in using this as potentially a biomarker of patients that would benefit from some sort of senolytic therapy. And so what we did was we had T cells from donors or patients that we edited to knock out that T cell receptor and then express our UPAR targeting chimeric antigen receptor. And so those are the blue cells. What we notice, and this is, we got lucky here, is that when we have cells that don't go through editing, but go through some of the processing to do CRISPR, they end up turning on UPAR within the culture. And what this ends up triggering is a set of kind of self-purification attributes of these products. And so once you make the CRISPR cell, it recognizes any of the unedited cells or any of the other senescent cells that might be coming from manufacturing. And eventually you get a product at the end that's 100% CAR T cells. And so some of the nuances of the signaling I don't have time to go into, but really it is the important kind of knocking out of the T cell receptor signaling that helps us drive this self-purification process. And so some of the data here is that within 10 days here, if you look at day 7 and day 17 of these cultures, you have, let's say, about a quarter of cells that are senescent that goes down to almost 0, 1.5%. And in the meantime, your CAR positive cells goes almost to 100%. And so we did this with a control where our CAR only recognizes a murine form of UPAR, and therefore any human cell would not be recognized even if it's senescent. And so that's this differently shaped receptor in this cartoon. And when we do the same exact CRISPR editing, and it's only a few amino acids that are different in our binding domain, we see this self-purification that seems to go up to 100% only happen when we have the human targeting UPAR. And so what this allows us to do is make a lot of cells really quickly that are really pure. And we're excited to now see how these would work in vivo. These work really well in vitro. We've taken human fibroblasts and irradiated them to make them senescent and then added our CAR T cells. We've also engineered a line to express the human form of UPAR. And when we add our cells, either at various doses, that's represented at 10 to 1, 5 to 1, 1 to 1, or our control cells, we see very high cytotoxicity dependent on our CAR. And then we also see similar effects against human fibroblasts, against microglia, as well as almost any cell that we've tested thus far. All right, so I'll finish up here and say that the first story, I think we have some very interesting ways to inject CRISPR into the brain. We're trying to improve delivery and also move these and scale up into formulations that could help start an IND. And we are also interested in understanding how this editing affects microglia. And in our second story, I hope I convinced you that we can make these interesting cells that can target senescent cells. Whether they would work inside the brain is something that we're exploring. We're also working with a number of companies, both here in the US and abroad, to think about how to scale up and manufacture these cells at higher efficiencies and higher purities. And we're also interested in applications outside of Alzheimer's, where aging or senescence is a big factor. And the most important slide here is the team that has helped us move all of this forward. Notably, Kirsten helped with the single nuclei analysis and our work with Subhajit. Lauren and David have posters that I put up here that you can see outside and have driven the T cell work. And we've had very important help from Brett Alston at UCSD. And certainly want to thank the help from the center, as well as a number of training programs and support from NIH to propel this work. Happy to take any questions.
And I think we only have time for one question, right, Cheryl? One?
Great presentation. This is amazing stuff. I think it's gonna, do wonders for the field. And, even into like FTD and all the genetic mutations in the related dementias. With the CAR T cell therapy, they do it at UW for some like lymphomas and cancer treatments. And a lot of times there's that cytokine response syndrome and the neurotoxicity. Are you guys seeing that with these therapies for the Alzheimer's treatments?
Right. So to be clear, all of the treatments for Alzheimer's that I showed are preclinical work, so we haven't dosed any patients. However, I think the dosing of CAR T cells, usually peripherally, through intravenous infusion, for lymphomas and leukemias, there's certainly a cytokine release syndrome that initially was thought to be almost devastating. I mean, the patients were in comas, but they were able to add an IL-6 inhibitor and at least manage that short-term cytokine release syndrome for some of these products. And so that's been clinically managed fairly well. I think one of the reasons why some of those initial CARs had a kind of syndrome and activation in the brain is that CD19, the target antigen, is expressed in neural cells in the brain to some extent. On one hand, it illustrates that T cells can get into the brain, right? So that is in some ways beneficial for us if we wanted to try to do this through a easier intravenous infusion. I think the glioblastoma trial and also a trial out of Stanford that did diffuse pontine gliomas have been able to manage some of the cytokine release effects in the brain. One of the nice things about CAR T cell therapy is that you can dose escalate. And so there is a way to perhaps push it to and individually manage this type of toxicity.
Thank you, Dr. Saha, for sharing that incredible work that you're doing. And if there are additional questions, we will send people to the posters of your trainees or come visit you on the 4th floor.